So, same way, yes.
We're here to investigate kind of the early stages of Earth.
In such a diverse planet that we have in terms of microbiology, why are there only such a selected few that can be able to live in these systems when all the others can't?
Such kind of ecosystem could be strongly related with the origin of life problem.
Here in these mud volcanoes we have carbons which are generated by geochemical reactions, and so I want to see if and how microbes can make a living or gain energy out of these carbons.
The serpentinization is a water and rock reaction. Serpentinization reaction can make organic carbon from inorganic carbon. So the organic carbon is really important molecule for microbes to live.
In these systems, Mariana Forearc serpentinite seamount, the pH of fluid become very alkaline, and sometimes over 12 point pH 12.5.
What we've seen down there is anaerobic methane oxidation and what we think is sulfate reduction. So we're trying to understand more about why those are the only games in town, so to speak, in terms of microbial metabolisms. And we're trying to understand a little bit more about the microorganisms that are actually able to pull that off at such a high pH. We have to realize that we're right on the boundary of what's habitable.
These subsurfaces are a lot different because there's so many factors about them that can make microbial life difficult. So first of all, there's the high pressure, there's the cold temperatures, it's high pH, and they're typically anaerobic. So all of these factors combined, and these organisms are still able to survive. It's kind of crazy when you think about it.
If you think microbiology, you think bacteria. But archaea, which is a very divergent, entirely different form of microbe, is the key player in this particular biosphere.
Archaea are really cool, and they're more related to us than bacteria are. And those are the ones that are able to pull this off, being able to live here.
Now we have many newly developed methods.
Metagenomics and metatranscriptomics and single cell genomics and all the other omicses.
And now we can identify the 150 microbial guides in just the water we can detect.
Molecular biology techniques let us be able to put communities back together again. It's kind of like Humpty Dumpty, but we're putting them back together again without having to grow these microorganisms.
We have serpentinization processes which have been going on for millions or billions of years, and so some people believe that life on Earth also could have developed in a similar environment, or that also life on other planets is driven by similar processes like here.
The serpentinization occurred many places in our solar system and even in outer solar system. So probably the extraterrestrial life can be present only at around serpentinization through the circulation system in our universe.
So if we can find microbes that are being able to survive and make a living at this extreme conditions, then they might be able to be an analog for microbes elsewhere or for the early Earth.
It's definitely an interesting place to look at where we come from and maybe where we are going to in the future.
And being out here with the team of microbiologists that we have is fantastic. I feel like I'm out here with the varsity team. It's exciting to be able to be a part of that.
I guess at the current state of science we would be more likely to return from the bottom of the Mariana Trench than from Mars. So probably this would be my choice.
Mars, duh. Elon Musk has taken us there. We're gonna build a new planet. And I want to see the blue marble.
Really hard to choose, but if I have a chance and if I can return, I want to go on Mars.
Both. Actually, I like to do both.
Yeah.
now I'm making the new manned submersible that can dive to the Mariana Trench.
Really?
Yes. And then now I'm leading some Japanese and some international project to send the spacecraft to the Enceladus, the Saturn's moon. But anyway, sorry, very long story.